My name is Ankita Jaykumar, and today I'll be presenting on Leigh syndrome. Leigh syndrome, also known as Leigh disease or as acute necrotizing encephalomyelopathy, is a rare mitochondrial disease named after Archibald Dennis Leigh, who first described it in 1951. In this presentation, you will also hear me refer to it by its abbreviation, LS. My aim today is to briefly go over the clinical presentation of LS and its diagnosis, then dive into its associated genes and inheritable mutations, how they impact cell function, and finally, what we know about treating Leigh syndrome. Leigh syndrome is an inherited disorder, and it affects approximately 1 in 40,000 live births. Although it is considered a rare disorder overall, it is one of the most common mitochondrial diseases. In affected individuals, signs of LS are typically apparent in infancy and at the latest by age 2, although there have been rare instances of LS onset in teens and adults. Symptoms of LS include a broad range of neurological issues, including seizures, developmental delay and regression, and resulting hypotonia, ataxia, dystonia, respiratory distress, feeding difficulties, and ophthalmologic abnormalities as well. The diseases in the same class as LS, the mitochondrial disorders, disrupt cell metabolism and energy production. The brain is one of the most energy demanding organs in the body, along with the heart, other muscles, and eyes, so the detrimental effects of disruption to ATP supply are most evident in the nervous system and muscle function. And that is the connection to the physical symptoms that LS individuals experience. Leigh syndrome is diagnosed by assessing clinical signs in combination with neuroimaging and laboratory testing. MRI scans of an LS individual, as seen here, will exhibit bilateral symmetrical lesions indicating necrosis, gliosis, or spongiform change in the basal ganglia, brainstem, thalamus, and periaqueductal regions of the brain. Blood or cerebrospinal fluids with metabolic markers, especially high levels of lactate, also support an LS diagnosis. Genetic testing is as easy as a cheek swab and is becoming more affordable each year. Although it's not possible in all LS individuals, when it is fruitful, it allows doctors to identify the type of gene mutation causing LS. Some genetic variants are also more difficult to identify with just regular genetic testing and may require sending out a muscle biopsy for testing. Leigh syndrome can be a devastating diagnosis, as parents of children with Leigh's disease are often given the prognosis that their child is only expected to live until 3 to 5 years old. This is a video of Abby McCurtain and her parents Greg and Heidi touching on that concept. The McCurtains have been advocates for LS research and support for children with special needs ever since Abby was diagnosed at 10 months old.
We were told by the other families, because one of them did lose their son, that if you have all that stuff planned out in advance, it makes it so much better, because you're not going to look back and say, we would have done X, Y, and Z, but you're just so emotionally so she said if you had everything, organized as much as you could. So I think we were just thinking, okay, well, Abby's 4, she's getting close to that 5, most kids wouldn't live past 5, we just didn't know what to expect. So I think we were just preparing for the worst and embracing the present.
Leigh syndrome can be inherited through autosomal recessive inheritance, and less commonly through mitochondrial and X-linked inheritance. While a de novo or spontaneous mutation affecting mitochondrial DNA is possible and is more likely in mitochondrial DNA than in nuclear DNA, this is extremely rare. Autosomal recessive inheritance is the most common pattern resulting in the conception of a child with LS. Both parents must be carriers of a mutant copy of a gene, and there is a 25% chance that their child will receive both copies, resulting in LS and the expression of the high level of mitochondrial dysfunction and neurometabolic disorder that characterizes the LS phenotype. In mitochondrial inheritance, also known as maternal inheritance, since mitochondria are only passed on through the mother's egg, the mother herself may be somewhat affected or not affected at all by diminished mitochondrial function. Mitochondrial synthesis is partially encoded in nuclear DNA and partially encoded in the DNA contained within the individual mitochondria, called mtDNA. As mitochondria replicate, some of them may gain mutations in mtDNA that change their metabolic performance. A child that inherits some mutated mitochondria is likely to have a higher ratio of unhealthy to healthy mitochondria, known as heteroplasmy. Individuals with a high level of mitochondrial heteroplasmy will express a metabolic disorder. In LS, over 95% of mitochondria are typically unhealthy. Some genes associated with LS are located on the X chromosome. Female parents and children who have 2 copies of the X chromosome, one from each parent, while male children receive 1 copy from their mother only. A carrier mother with 1 X chromosome expressing the defective gene may or may not show some signs of a metabolic disorder. She has a 50% chance of passing on the affected copy to her child. Like their mother, female children have a 2nd X chromosome with normal gene expression and may or may not be somewhat affected, depending on which cells have turned on which X chromosome. A male child who receives a pathogenic copy does not have another X chromosome to offset, so he will have LS. Lastly, a very rare inheritance pattern for LS, autosomal dominant, has been observed in only one gene, DNM1L, which codes for a GTPase involved in mitochondrial cell division and embryonic development. A single defective copy from either parent will lead to LS, with 50% chance of an LS child if one parent is a carrier and 75% if both parents are carriers. And you may notice that I only outlined the inheritance patterns of carrier parents and not affected parents with LS passing on mutant alleles to children. This is due to the life-limiting nature of LS and the unlikelihood of an LS individual living to reproductive maturity or being able to conceive a child. Since every cell in the body, except red blood cells, have mitochondria, they are all affected by primary mitochondrial disorders like LS. The number of genes associated with LS mitochondrial health and respiration number over 110, with new ones continuing to be added to the list. Most of these genes encode proteins that function in 5 complexes within the electron transport chain, or ETC, at the inner membrane of mitochondria. Mutations leading to deficiencies in complexes 1 and 4, certain mitochondrial DNA, and in the pyruvate dehydrogenase complex are the most common causes of LS. Here are some of the main genes associated with the development of Leigh syndrome and the typical location where point mutations or deletions are seen in LS. NDUS4 is a nuclear gene which, along with the mitochondrial gene MTND5 and many other NDUF and MT families, encode ETC complex 1 subunit proteins. SIRF1 encodes a protein in the assembly pathway for ETC complex 4, and mutations in this gene are the most common nuclear genetic cause of LS. MTATP6 encodes the ETC complex 5 ATP synthase subunit 6. And PDHA1 is a sex-linked gene located on the short arm of the X chromosome and encodes a protein in the pyruvate dehydrogenase complex that supplies the citric acid cycle. The common theme with all these genes is the role they play in providing the code to make proteins that are necessary for oxidative phosphorylation and ATP production. Any deficiency of mitochondrial respiratory chain enzymes or upstream components necessary to generate the electrons to make ATP through cell respiration can disrupt cellular energy production. I touched on the genes involved in LS, and now I'll elaborate on the proteins they code for and how defects in those proteins affect various pathways leading to ATP production. NDUS4 mutations result in a defective NADH ubiquinone oxidoreductase subunit, also known as NADH dehydrogenase subunit 4. The whole enzyme catalyzes electron transfer from NADH to the coenzyme Q10, or ubiquinone, and moves proteins across the inner mitochondrial membrane to build the gradient used by ATP synthase to make ATP. Induced 4 mutations inhibit this electron transfer and disrupt the electron transport chain. They also appear to be associated with early onset and death in LS. Similarly, SIRF1 mutations disrupt electron transport through complex 1. It codes for a protein involved in regulating the assembly of complex 4, also called the cytochrome C oxidase complex. A defect in this protein leads to deficiency in complex 4 of the ETC and ultimately ATP synthase via oxidative phosphorylation. PDA1 codes for a subunit of the pyruvate dehydrogenase complex, which decarboxylates pyruvate to acetyl-CoA, which is needed to initiate the citric acid cycle. Without it, the cell is forced to rely on glycolysis rather than oxidative phosphorylation, resulting in limited ATP production and lactic acid buildup. Since the body is also able to obtain acetyl-CoA through the breakdown of fatty acids, LS individuals with this mutation may have some symptom relief on a high-fat, low-carbohydrate diet. Lastly, we have the 2 mitochondrial genes most implicated in LS. MT-ATP6 encodes a subunit of ATP synthase, also known as complex 5, in the ETC. As we already know, this complex is the final step in the chain and generates most of the normally functioning cell's ATP. MT-ND5, like NDUS4, codes for a subunit of NADH dehydrogenase. A defect in ND5 will similarly disrupt the transfer of electrons from NADH to ubiquinone and the cell's ability to generate ATP, which is the common final result of dysfunctions in components of any of these pathways. There is still no cure for Leigh syndrome, but there are treatments that can improve the quality of life for LS individuals and sometimes increase lifespan. Genetic counseling and screening can help individuals with a family history of LS make informed decisions about conception and pregnancy. Families with an LS child can also benefit from palliative care consultations at the point of diagnosis to help them understand all their options for improving quality of life, such as a gastrostomy tube, vaccination protocols and infection protection, at-home care, and to provide resources for emotional aspects such as anticipatory grief. A very small number of children have a specific mutation on the SLC19A3 or BTD genes that lead to deficiencies specifically in B vitamins or their cell transporters, and they may benefit from supplements of thiamine and biotin. PDHA dysfunction can sometimes be managed with thiamine and a ketogenic diet, as we discussed earlier. The Cure Mito Foundation is working with the University of Texas Southwestern on preclinical research for virus-facilitated gene therapies targeting SIRF1 and MT-ATP6 mutations that cause LS. And a study this year by Puigdemont et al. Demonstrated that cannabidiol reduces LS neurodegenerative and motor decline symptoms in mouse models and improves cellular function in LS human fibroblast cells. An even more promising pharmaceutical development for LS treatment is the drug EPI-743. A clinical trial study of EPI-743 therapy in 10 children initially with genetically confirmed Leigh syndrome showed stabilization and even reversal of disease progression, and that research has since expanded to many sites. EPI-743 and another drug, RP-103, are thought to improve the redox imbalance caused by high levels of reactive oxygen and nitrogen species caused by mitochondrial respiratory chain dysfunction. They do this by increasing intercellular glutathione, an antioxidant that helps defend cells from free radicals. Both drugs are currently under investigation in multiple clinical trials, with SIRF1-affected individuals much more likely to be entered into newer trials for EPI-743. Although the trial periods for the drugs span only about 13 weeks, trial subjects that had positive results could consult with their physicians and extend their time on the drug. In a previous slide, we saw Abby McCurtain at age 4, with her mom explaining that she was almost 5, an age to which many LS individuals do not survive. Abby entered into a clinical trial for EPI-743 shortly after that video. This is a video of Abby at age 10 playing with her younger brother, Thomas, who does not have LS. And then a photo here of her from this year on her first day of 8th grade at 13 years old. While the drug is not curative and LS is still a life-limiting disease, it continues to help relieve symptoms and extend her life over a decade past the initial expectancy.
You got it, sweetie.
Abby, you can do it! Abby, follow your
Yay!